Great. Well, thanks, Bradley, for that introduction. I'd forgotten about that, actually, but thanks for reminding me. I'd just like to start by saying it's great to be here. In particular, great timing with Tony's recent knighthood. And Tony, who would have thought that, we joined the Gurdon Institute 31 or 32 years ago as junior group leaders. And who would have thought then that we might both be Knights of the Realm at this stage? I mean, maybe John and Ron did when they were hiring us, but those times were great. It was friendly, often fierce but friendly competition and benchmarking between our groups, which I think drove ourselves on, but also members of our group. And I remember very well various members of his group, as well as my own, of course, including John Milner, going around with ice buckets and talking to members of my group how they'd best like to have a company that could suit their antibody needs. The rest is history. So those times were great. I've now moved on to the CRUK Cambridge Institute. The Gurdons come up various times. It was a fantastic place. It is still a great place to do science. Lots of interesting memories, lots of great parties. And so I just thought it'd be quite interesting to bring out some photos of those. So this one here, I think was about 7 years ago. That's me and Tony, if you don't recognize us. This was more like 17 years ago at a Christmas party. And I was actually looking for photos from earlier parties, and I found some, but I decided that they weren't really suitable for sharing to an audience like this. But, maybe Tony and I or Nicky Mann or somebody will be able to share those with you at some other stage. So where I am right now is at the CRUK Cambridge Institute on the biomedical campus, but still great connections to the Gurdon. This is my lab on retreat in Norfolk last week. Tomorrow we're going to glorious Molar Centre for our this year's retreat, just up the road. So my lab stumbled into the area of DNA repair many years ago now. It's chance observation that took my lab from a transcription lab into the DNA repair arena. And just to share with you the fact, there's not many aficionados in the audience, that our DNA is continually being bombarded by chemicals and other agents, some from the outside, but much of this from endogenous sources. So every human cell at rest, away from radiation sources, is sustaining around 100,000 DNA lesions per day. And when you think you've got around 5 times 10 to the 12 cells, that's a lot of DNA damage going on in your cells. I said last week, if you stood outside a sun-exposed cell, every sun-exposed cell was being damaged about 100,000 times an hour. That's not taking place today, but it was last week. So we have many different DNA damaging agents, many forms of DNA damage, and they're repaired by different DNA repair systems, which are dedicated to different forms of DNA damage. And in some cases, the same lesion is repaired by multiple pathways, which is leading us into areas where we're considering, is the cell using a certain pathway? Well, we know this is using a pathway for, let's say, double-strand break repair in certain contexts, preferentially to others, for good reason. Okay. So over the years, we've learned that cellular responses to DNA damages is very important for understanding cell biology and with very strong connections to human health and disease. With DNA damage, intrinsic or acquired defects in cellular DNA damage responses underpinning a whole range of conditions, some of which are shown here. So in broad sense, the way cells respond to DNA damage is there are DNA damage sensor proteins, including a protein called Ku, which is the discovery of the DNA end-binding protein Ku and its connection to a DNA break activated protein kinase. That's what my whole career in DNA repair was focused on. So my whole DNA repair career is based on what Ku does, hence the name for my first company, Kudos, I'll share with you in a few moments. So DNA damage is recognized by proteins. These are influenced by other things, such as nuclear architecture, chromatin structure, etc. And then these DNA repair proteins and associated factors engage in the DNA repair mechanisms themselves, but also trigger various signaling pathways that can do things such as slow or change cell cycle progression, cause cells to go into senescence or program cell death mechanisms, or mediate transcription or post-translational modification cascades. So over the years, we've learned that these very intricate DNA damage response processes are not just individual proteins or pathways, it's more of a network of interactions and positive and negative reinforcements going across all of these different components. And many of these processes are controlled by reversible post-translational modifications ranging from phosphorylation, ubiquitination, sumoylation, and poly-ADP ribosylation. So we now know that there's actually probably closer to 2,000 human proteins with strong connections to these DNA damage response processes. But there are undoubtedly additional components that remain to be identified. And what we're increasingly seeing in my lab is that if we carry out CRISPR screens, for example, in sensitized backgrounds where we've already blocked one pathway, we're able to start uncovering what might look like backup pathways, but are actually more like specialized pathways that are used in different contexts. One area that my lab is very focused on, and many other labs in this arena, including, I think, collaborators of Todd from what he was talking about, is to understand connections between different pathways, look at different outputs from different screens and different drugs, and understand those connections. So this landscape of interactions and regulation across this landscape is something that is still very much largely uncharted. Another thing that we've learned over the last couple of decades or so is that many proteins involved in cellular responses to DNA damage are enzymes, and on paper at least druggable. And it was with that in mind, with help from the Cambridge University Translation Office and Cancer Research UK, that I set up Kudos a number of years ago. It turned out that it was I guess it was the world's first drug discovery company. I couldn't get pharma interested at that stage, and I couldn't get VCs interested initially, but I did get several VCs interested. And it was rather interesting. At the weekend, I was presenting prizes and giving a talk at Speech Day at a school in Hertfordshire. And it turns out I was invited there by a VC who was part of the original investors in Kudos. And she introduced me to her mother, who was a super responder to a drug that this company, Kudos, developed, olaparib, which was a very nice afternoon for me. And I think it was nice meeting her. So one of the things that really did it with Kudos in terms of the business plan and some of our initial studies were basically bringing together these two concepts, that many cancers lose certain aspects of cellular response to DNA damage. They get mutations or epigenetically deregulate certain DNA repair pathways and components. And mapping that together with this concept here, synthetic lethality, a concept synthetic, you're bringing together more than one thing together to generate something new. In this case, a cell or an organism can tolerate loss of one gene or a change in one gene. It can tolerate loss or change in another gene and remain viable. But if you bring these two genetic changes together, that can give rise to cell death. And so the concept, and this was in the original 1997 Kudos business plan, was basically to translate this concept, genetic concept, couple it together with this knowledge into an opportunity to selectively kill cancer cells. In this case, a cancer cell which has lost pathway B is going to be heavily reliant on gene A, and the product of gene A much more reliant than the normal cell that might have this synthetic lethal arrangement. So to cut a long story short, Kudos developed this drug, formulated it, and actually took it into the first patient. So AZ did acquire a drug. We'd even show them how to take it to the clinic, to the right patients. It took a while. Clinical trials aren't trivial, and it costs a lot of money. And AstraZeneca stuck at it, and eventually this drug was actually approved. And I'll tell you very briefly about that in a moment. But what olaparib does, it's a small molecule inhibitor of a DNA damage-activated enzyme called poly-ADP ribose polymerase. It goes in two forms, PARP1 and PARP2. So PARP is a DNA damage-activated enzyme. It recognizes DNA strand breaks. This activates its catalytic activity, and then it mediates the poly-ADP ribosylation of itself and other components on chromatin to modulate chromatin structure and help bring in the latter stages of the of the base excision repair and single-strand break repair machineries. So if you inhibit PARP via drugs such as olaparib, you slow down single-strand break repair, you accumulate these kind of lesions, and in most cases, you also end up increasing the resident time of PARP on DNA damage, because its auto-ADP ribosylation is normally required to trigger its dissociation to allow the latter repair components to come in. So PARP inhibitors such as this slow down repair, and they also end up trapping PARP on DNA. Now, it turns out that this is not particularly toxic for most cells. Olaparib and other PARP inhibitors, such as GSK's niraparib, get metabolized, excreted, and whatever turned over time, and these single-strand breaks are repaired. But these single-strand breaks and blocked PARP here are much more toxic to cells in S phase. A cell going through replication, encountering those kind of lesions, you could imagine how those breaks are going to lead to double-strand breaks or to replication fork collapse. But it turns out that normal cells actually can cope with those replication-associated DNA damage events in the presence of PARP inhibitors very well, because they are very readily repaired by a range of mechanisms collectively called homologous recombination, which are controlled by the actions of proteins such as BRCA1 and BRCA2. So normal cells, normal human cells or other mammalian cells are fairly insensitive acutely to treatment with PARP inhibitors such as olaparib. However, if a cell lacks BRCA1 or BRCA2 function or other components of the homologous recombination machinery, we learn that they're very, very sensitive to those kind of agents. And this goes back quite a number of years ago. This is a collaboration between my lab, QDOS, and the lab of Alan Ashworth, then at the Institute of Cancer Research. He had some paired cell lines, deficient or proficient in BRCA1 and BRCA2, and we had drugs targeting enzymes such as PARP. And the striking results, which we actually got before an MTA was even in place, I'm not sure you can actually do these, or companies would do these kind of things now, are these data here showing that there's a strikingly selective effect of this PARP inhibitor on killing cells with BRCA1, or in this case, BRCA2 deficiency. These are actually mouse ES cells, the initial experiment. This phenotype spans across a range of different types of cells, across genetic backgrounds, and across species. So BRCA2 deficient cells are much more sensitive. This is a log plot in each case. This is log surviving fraction of cells, a log plot of increasing PARP inhibitor along the bottom. And this really excited us because and also the group of Thomas Halliday and Nicola Curtin, who published similar findings at the same time in a back-to-back scenario in Nature, that this agent and similar agents might be selectively toxic to the cancer cells in BRCA1 and BRCA2 mutant carrier people, but not the normal cells of those patients. That's because BRCA carriers inherit just one bad copy of BRCA1 or BRCA2. They have one wild-type copy and one bad copy. And it's loss or downregulation, epigenetic or otherwise, of the wild-type copy that's invariably connected to tumorigenesis in those patients. So to cut a long story short, it was a long story, but it was a very successful story in the end. AstraZeneca brought this one over the line with clinicians, and more latterly, olaparib was partnered with Merck, who have taken this drug forward. So in 2014, quite a number of years ago now, this became the first approved DNA repair enzyme inhibitor drug. It's the world's first synthetic lethality medicine, if you want to call it that. It's the first cancer drug, as far as I'm aware, targeting inherited predisposition, although we know that PARP inhibitors like olaparib also work in what we call sporadic cancer settings, where BRCA1, BRCA2, or other components of the homologous recombination apparatus are lost somatically. It was notable that this type of agent, PARP inhibitors, are being used and in many cases launched with a genetic diagnostic, so targeting the right kind of patients that are most likely to benefit from the agent. And for olaparib, and actually for a range of other PARP inhibitors now, these are approved across a range of different cancers. And it's rather nice to learn that for everybody connected to this over the years, that over 150,000 patients now have received olaparib, and this is actually making Merck and AstraZeneca quite a bit of money. I should point out that, and I'll never do this again, if my company is acquired by a pharma, no milestones and no royalties. Anyway, that's a learning experience, not just for me, but everybody else, Cambridge Enterprise and the VCs. Okay, let's move on. What it really matters, of course, is the patient benefit. And not all patients respond well or even respond, but a number do, often for many, many years. And just a few years, I met this lady in the Cambridge Botanic Garden. It was about this time of the year, but it was 33 degrees centigrade at the time. I remember it very well. And this is a lady who was a super responder, and about a decade on after having terminal ovarian cancer was still more or less, well, was cancer-free, as detectably cancer-free. So where we are now more broadly with PARP inhibitors, and there are 5 now approved PARP inhibitors helping patients across the globe, is that as they are being used earlier, the benefit is generally proving to be more substantial. For example, in the SOLOS trial here, you don't need to be a clinician or a statistician to see that there's a big difference between the control and treatment arms here in terms of progression-free survival. So obviously there's a lot of excitement that patients on PARP inhibitors such as olaparib are faring better than they would have done otherwise, and that many of these patients have very durable responses and perhaps cures. On a negative side, however, you can look at this and say, well, okay, even cancers with BRCA mutations that maybe initially are responding to PARP inhibitors nevertheless evolve resistance. So there is a great challenge, I think, to understand resistance mechanisms and maybe try to even exploit them. There are various mechanisms now known. One mechanism is reversion mutations in BRCA1 and BRCA2, which bring back at least partial BRCA function. But that is driven by DNA repair mechanisms. So there is some hope that if you can intervene in those DNA repair mechanisms generating the reversions, you might be able to start heading off that kind of reversion mechanism. There are other resistance mechanisms we and others have discovered, in some cases through hypothesis-generated experiments, in other cases, and actually more successfully, through genome-wide and focused CRISPR screens, asking the question, in the DNA repair arena, if a BRCA1 deficient cancer cell is very sensitive to PARP inhibitor, can I identify other genetic changes that could give rise to resistance? And that takes us into a very exciting area for DNA repair biology, is that cells, unfortunately cancer cells, there are various ways to rewire the DNA repair apparatus that allows you to get around, for example, BRCA1 deficiency. So what we and many groups in the world are sort of doing now are using CRISPR screens to try and understand resistance mechanisms. And optimistically, maybe overly optimistically, but that's part of the way I am, what we're increasingly finding is that if you find a resistance mechanism to one drug, such as, for example, resistance to olaparib in the context of BRCA1 deficiency, that resistance cell, if it's rewired the DNA repair processes to give resistance to that drug, invariably that drug turns out to be more sensitive to another DNA damaging agent, such as ionizing radiation or maybe DNA protein cross-linkers. So hopefully, these kind of approaches, at the very least, will give us some further insights into DNA repair mechanisms, might provide opportunities that hopefully one day will be monitorable in patients and maybe exploitable in real time over the course of their cancer trajectory. I think it's also very exciting to think that many cancer drugs, including PARP inhibitors, might even be used prophylactically if the right individuals can be identified at the right stage, potentially even precancerous, to wipe out those precancerous cells before they've even evolved into a fully blown malignant state. So with that as a bit of history, what an area that my lab is focusing on quite a lot now, in addition to looking at new DNA repair enzyme inhibitors, we're also thinking about some conventional chemotherapeutics, including DNA hypermethylating agents. And just to give you a flavor of some of the projects in the lab ongoing now, funded through Cancer Research UK RadNet, Aldo Beda and Eugenia Arrieta-Sanz in my lab are looking at drivers of sensitivity and resistance to radiation, identifying many of the known key players, but also identifying other players. And this is actually also taken us not just into the DNA repair area, but also taking us into other areas. Radiation, of course, doesn't just damage DNA, it also damages RNA, proteins, and so some of the resistance and sensitivity hits are mapping into those spaces, and then we're exploring those in more detail. We're also looking at other agents such as etoposide, a double-strand break generating enzyme through inhibiting topoisomerase II, as well as other agents such as camptothecin and topotecan, which target TOP1. And that's taking us to identify new players in the DNA damage response, as well as identifying potential resistance mechanisms that might one day be useful in the clinic, including a newly identified factor, RAD54L2, that we're focusing on. And I'm not going to go into the details, but we can discuss them afterwards. We also are profiling a range of different agents, including decitabine and other related agents, across a range of different genetic backgrounds. And our conclusion is that, yeah, hypermethylation is a key component, at least in some settings, but DNA and actually RNA damage and actually ribosome stalling might be also contributing quite substantially to toxicity, at least in culture. Another arena that I think is going to come up quite a bit, and I know this is an area that the Milner Institute has put a huge amount of focus behind with Namshit Han and his colleagues, is computational platforms, which of course you need to interpret things such as CRISPR screens and other omic data, but also AI and machine learning. So recently, in fact, a couple of years ago, my lab launched DDR-CS, which is an interactive platform which allows you to look at all published CRISPR screens in the DNA repair arena in various ways. It's turning out to be quite popular. It's been hit over 300,000 times, this site, as well as more recently using AI and machine learning approaches. Tran, a postdoc in the lab, has recently basically thrown into his black box of AI machine learning all the internal as well as external DNA damage repair focused CRISPR screens. And I wasn't convinced at all about this approach at the outset, but when Tran came into my lab with various clusters that are thrown out through this, what I'm calling a black box approach, there are certain clusters that everybody in the lab got excited about because they have proteins and components and genes that we know about, as well as some potentially new ones. But I was actually more interested in one that didn't demonstrate any interest at the outset. I'm not showing it here, cluster 5, which doesn't have any of the factors that we're used to thinking about. And actually, most of these factors are connected to mitochondrial biology. So I think I'm very excited about these type of approaches, which are taking us into areas not just where we're used to looking, but also suggesting that we start looking into new dimensions. Okay, just to round off then, I just want to highlight that, of course, a big focus in labs like mine, because of strong connections between DNA repair and cancer biology, is thinking about cancer. But as I pointed out earlier on, cellular responses to DNA damage also impact on a range of other diseases. And of course, biology in general, health is about balance and disease is about imbalance. So as is the case for cancer, if we can understand the molecular balances that connect to health and the molecular imbalances that underpin disease, we're going to gain insights into disease biology, and hopefully this will point to new diagnostic, prognostic, and hopefully therapeutic opportunities. And so with that in mind, I was the prime founder for a company called Milner Therapeutics, which is operating on the Cambridge Babraham Biomedical Campus at the moment. The focus of Mission is being to target a chemistry program on a set of enzymes called deubiquitinating enzymes, a very difficult to drug set of enzymes. But we've established a platform in Mission that allows us to develop pretty potent compounds and selective compounds across many of the cysteinol type DUBs. And several of those are now moving forward into preclinical and 2 of them into clinical studies. This one here, just recently this year, we announced moving MTX-325, a small molecule covalent reversible inhibitor of USP30, a DUB, into actually a couple of different indications, but here for Parkinson's disease. And the idea behind this drug is to rebalance mitophagy, which in certain situations in Parkinson's gets imbalanced through genetic or other factors. And this is quite meaningful for me because my mum died with this terrible disease just a few years ago. And then finally, a few years ago, incubated in the Gurdon, yet again, was this company, Adrestia Therapeutics. And the focus of Adrestia, which was funded by RN Innovation Capital, and actually some substantial funding that we got from GSK, championed by Tony Wood, was to flip things around and think, okay, DNA repair is connected to many diseases. We've already heard about this synthetic lethality opportunity where you can take advantage of a genetic or other weakness in a cancer cell to push it over the edge into death. Synthetic lethality is one extreme of genetic interaction. The other extreme of genetic interaction is genetic suppression, synthetic rescue. And in this scenario, synthetic rescue, a phenotype or change caused by a change in one gene can be rebalanced, offset, restored by changing the product or the gene, the product of another gene or the gene itself. So the concept behind Adrestia was synthetic rescue. So as opposed to synthetic lethality drugs, our idea is to set up a platform where we can identify opportunities to rescue genetic diseases. So often we start out with a rare, relatively rare genetic disease, but with a mindset that that rare genetic disease might unpick a mechanism which is also evident in sporadic disease. So we've, over the last few years, we've developed that platform and have several projects moving forward. We were looking for VC funding until quite recently, but instead, and I think it was a great decision by the board, we went with an acquisition offer from Insamed Innovation UK, which has recently announced fantastic clinical data from one of its clinical programs and is now growing into a substantial pharmaceutical company. So what was addressed here is now Insamed Innovation UK, and later on this year is going to be moving into newer, larger space on the Babraham Science Campus. So I'll leave it there. Thanks.
Thank you. Any questions? And again, online, if you have any questions. I have one, if that's okay. You have a drug on the market that's got your fingerprints on it, 3 spin-outs, 2 of which were successfully acquired. What's next on the bucket list?
Not to set up another company. Hopefully. Engage with those companies going forward. I'm still actively engaged with Mission and Adrestia. Maybe spend even more time in my academic lab. Not sure how many years I've got left, but hopefully quite a few. I'm an academic at heart, and that's what I want to do most of all.
Fantastic. If there are no questions, thank you very much. Oh, sorry. Sorry there. Just apologies. Any others?
I'm very small, it's fine.
Sorry.
So I'm just quite interested in synthetic lethalities in general. So obviously the PARP-BRCA interaction has been extremely exploitable. I don't know if you could talk to the challenges there and, you know, in terms of tox and, you know, whether you've seen, I guess it hasn't been so easy in other cases to really exploit synthetic lethality. So what is it about that interaction and what have been the challenges from a tox perspective?
No, great question. In the end, it's all about therapeutic window, and that's managing tox to an extent where you're efficacious, but not causing overt toxicity to the patient. And I think in the end, we were lucky with PARP inhibitors in the context of BRCA deficiency. It's not proving anywhere near as easy in other areas. I think there will be other successes in the future coming through. I think the issue is that the BRCA deficiency is fairly well hardwired, and it also operates, and I did point on this, across different cell types, across even different species. So this is a very robust relationship between those targets. And of course, in the case of cancer, you know, you can't just kill 90% of the cells. You've actually got to kill or stop growing all of them. So I think that's a big challenge, and do it in a situation where you're not causing overt toxicity. I'm actually optimistic that synthetic rescue might operate more broadly. Again, it's going to be about synthetic rescue, but in there, you probably don't need to correct all the cells. Even correcting a subset of the cells that were on track to dysfunction or die might be enough to alleviate the disease to a big degree.
Super. Okay. Any other burning questions? Just there.
Hi, Steve. On the resistance to olaparib, you talked there about you have to kill all the cancer cells. Do you think that resistance is coming from a stem-like population that already have distinct, we know that they may have distinct DNA repair pathways. Do you think that's where the resistance is coming from? Or do you think the resistance is coming from cells that were sensitive to olaparib and are now no longer sensitive?
No, that is a good question. I mean, this is outside of my domain of expertise. We use simple cell models. But the recent data are showing that these are probably right, because many PARP inhibitor treated patients, in many cases, at least in the past, and less so now, have been treated with other agents already. They're being treated with DNA damaging agents which generate mutations, and that is fueling various reversion as well as other mechanisms. So I don't know of any specific data that's saying from cancer stem cells or equivalent cells, but that's an area that is definitely worth exploring. Yeah.
Okay. Super. With that in mind, thank you very much indeed.
Thanks, Bradley.